Is a topic that we started to talk about right after evapotranspiration and before the first exam. And it's relevant to evapotranspiration because, of course, the water that is being evapotranspired is often coming from the soil. But it is also relevant to the next topic we're going to talk about, which is infiltration, because it influences the way that water is able to move vertically through the soil and potentially recharge groundwater or feed a stream. So, to remind you of some of the key definitions about soil water, we're going to define water content. We'll use the letter theta for that. And often we're talking about volumetric water content. So the volumetric water content in a soil is the volume of water over the total volume of that piece of soil. So if all of the pore spaces in the soil are full of water, it's saturated, and the volumetric water content is equal to the porosity, or the amount of pore space in the soil. We can also think about things in terms of how close to saturated they are. So the volumetric water content divided by the porosity defines a degree of saturation. So you've seen this slide before, but I wanted to remind you of that. So if all of the pore spaces are full, then it is saturated, and some of the water will drain vertically due to the effects of gravity. At some point, called field capacity, there will be a lot of water in the pore spaces, but there will also be some air, and water will no longer freely drain gravitationally. Some of that water that is in a soil then will be held as capillary water, and some of the water will be held as adsorbed water, so that is more tightly bound to the soil particles. When all of the capillary water has been used up, then we get to just the adsorbed water here, and we reach what's called the wilting point. So you've seen this slide before as well, but reminding you about the difference between adsorbed water, which is held to the soil by hydrogen bonds, and generally plants can't get at that. In slightly larger spaces between the grains, that's where you get the capillary water. And this is an example of two capillary tubes. The amount of capillary water we can store is a function of the size of the pores. So smaller pores allow more capillary rise, and if you get to really big pores, say up here, then that's where you're going to get the gravitational drainage happening. Okay, so field capacity then, remember that's where there's some air, there's no more gravity drainage, but there's a lot of capillary water and adsorbed water left. We can actually see that in soil moisture data. So this is a graph of water content as a percent versus time. This is in November and December. And you see the effects of some rain events here. So water content increases sharply. Maybe it stops raining and it starts to drain, and then water content goes up again, and then it starts to drain, but at some point it levels out. Now, it's relevant here that we're looking at late fall data because the plants aren't growing in this situation. So they're not evapotranspiring. It's not during the growing season. And so at some point, all of the water that's going to drain away has drained away, and we get this relatively flat line here. So at a half a meter depth, our water content stabilizes at about 0.25 or 25%, and that is the field capacity of the soil at that depth. At a somewhat deeper depth, our water content stabilizes at around 0.20, and that is the field capacity of the soil at that depth. So some important points here are that field capacity represents the stable water content that you get in a soil outside of the growing season, and that you can have different field capacities at different depths within a soil. So what would this look like if the plants were growing? Well, we wouldn't see a stabilization here because plants would be continuing to draw water up through their roots and transpire it out. So we would see some continued decline in water content in the soil if this were during the growing season. I think you've seen this slide before as well, but just to remind you that the field capacity, the water that can be held against gravity, and the wilting point, the bottom end of plant available water, vary with texture. So a loam is a soil that's a mixture of sand, silt, and clay, and that is usually considered a very good soil for growing crops in. So there's a big difference between field capacity and wilting point in the soil, so there's a lot of plant available water. If we go over to a clay soil, we have very small pores, so gravity drainage drops off pretty quickly because it's hard to move water through those small pores, and a lot of it gets held as capillary water. But there's also a lot of adsorbed water, so there's a much lower amount of available water in a clay soil. Similarly, in a sand soil, you have big pores, so there's a low amount of available water because the field capacity and the wilting point are both low. So these sort of mid-range textures that have a mixture of sand, silt, and clay give us the most available water and have traditionally been favored by farmers as the best place to grow most types of crops. So we're going to shift gears now, and we're going to talk about the ways that we measure water content. So we're going to start with a straight up measurement of water content, but that requires collecting a soil sample in the field and bringing it back to the lab. So this is good for one-time measurements in a small number of places, but won't give us continuous data over time, and it also doesn't give us a wide range of spatial data. Right, so what you're going to do is you're going to go out in the field and you are going to extract a soil sample or take a soil core and get the mass of that soil core as quickly as possible, ideally by bringing a balance into the field. If that's not possible, by quickly bagging it, keeping it on ice, and bringing it back to the lab, and then using a balance there to get mass. Once you've gotten that wet weight, you're going to put it in a drying oven, just warm enough to speed up evaporation from the soil. And you're going to dry that for 24 hours, weigh it, dry it for another 24 hours, weigh it again until you get to a constant mass, which would be the dry weight of the soil where all evaporation has already taken place. So then you're going to use the difference in the wet mass versus the dry mass over the dry mass to calculate what's called a gravitational water content. So notice that this is in terms of mass, whereas the definition I gave you a few minutes ago was talking about volumetric water content. So if you want to get volumetric water content, you also need to be able to move between mass and volume. So you're going to do the same thing, but you also need to know the density of the soil. So operationally, what this means is you need to know not just the mass of your soil, but you need to know the volume of your soil sample. So taking it in a defined coring unit is a great way to get bulk density. Once you've done that, you can take your gravitational water content times the density of the soil divided by the density of water, and you can go from gravitational water content to volumetric water content. All right, so as I said, that's great. This is the number one way to measure volumetric water content, but it's not applicable to get long time series of water content or getting lots of data all through a study area. So there are a number of methods that we can use in the field that will give us proxies for volumetric water content, and then we can often calibrate them to give us the volumetric water content directly. And the way we calibrate those proxy data is often by doing this, by collecting the soil sample, getting the gravitational and the volumetric water content, and then essentially correcting our proxy data for the volumetric water content at that time. Okay, so two of the most common methods for measuring water content in the field are time domain reflectometry, or TDR. That is what is pictured here. And these are considered sort of the number one best way to get good measurements of water content in the soil. A cheaper alternative that uses some of the same principles are what are called capacitance sensors. And the way both of these work slightly differently, but the general concept is that they're measuring the dielectric constant of the soil. That's how easily the soil can transmit or resist electricity. So if it's a dry soil, the dielectric constant will be in the range of about 3 to 5. If you just held these sensors up in the air, the constant would be 1. But water, as we know, is a really good conductor of electricity. That's why you get out of the lake or the pool during a thunderstorm. And so the dielectric constant for water is 80. So now if you can imagine, you stick these sensors into a soil mass that has some soil and some air and some water, it's going to vary someplace between 3 and 80. And how high that dielectric constant is going to be is going to depend on how wet the soil is. So we can measure the dielectric constant. We can attach our probes to data loggers, and so we can get measurements very frequently. Or we can use sort of a mobile version, which is what's shown here, and we can walk from place to place in the field and get one-time measurements. So, and we'll actually get a chance to try some capacitance sensors in the field on our field trip in a couple weeks. Now, we're measuring the dielectric constant. We want volumetric water content. A lot of sensors have built-in calibration, so they won't even tell you what the dielectric constant is. They will instead tell you what the volumetric water content is. And often that's pretty good. But if you want really accurate data, or if your soil is not a typical soil, then you go back to the previous slide, you take some soil cores, you also measure the dielectric constant at the same time, and you calibrate your data to the conditions of the soil that you're interested in. So we've used a number of capacitance sensors in our research. So this is a setup that we had at the Watershed Stewardship Center a few years ago. So we have 4 different capacitance sensors here, all connected back into a data logger here. And so you can see they sort of look like tuning forks. This is sort of a protective case right here that we'll take off, and then we'll jam these into the soil. We were putting these soil moisture sensors on a green roof, so it was a very shallow soil, so we just put them in from the surface. You can also dig a hole or a pit and then put them at various steps in the side of the pit. The way you deploy them, of course, depends on the information you want. So on the green roof, we were interested in downslope variations with the roof. So you can't see the soil moisture sensors once they're in the ground, but this is what you could see at the surface. So we had some PVC tubing to protect the cable from any animals that might be up on the roof. We did have a goose nesting on the roof for a while. I suppose you could get something like mice or anything that could get up to the roof, could cause a problem. We didn't want to bury the cable and dig a trench because this soil is only 7 centimeters deep, and we really would have destroyed a chunk of the roof. So this was our way of protecting our cables. And then at each of those verticals, you would have a soil moisture sensor buried at that location. So we were interested in potential downslope variation in soil moisture on a green roof. There are some things that we'd expect to see in a natural hill slope, where we would expect to see drier soils at the top of a slope in an upslope position and wetter soils in a downslope position. So we were curious whether we'd see the same thing in the green roof. And this is what the data looked like. So Laura Sugano is defending her master's thesis on Monday. You may be watching this video before then. I'm recording it before then, or you may watch it afterwards. But this is a slide. She'll show something very similar. So this is that green roof soil moisture data from that array of sensors I was just showing you. So the purple bars at the top represent rain intensity, and then the 4 colored time series represent soil moisture on the green roof starting in the spring before the growing season. So we can see some field capacity over here. And then we see rapid responses to rain events. And here in the summer, during this dry period, we don't see things stabilizing at field capacity, but we see the plants continuing to withdraw water. So here's a nice evapotranspiration signal in the soil moisture. Was there a common pattern in terms of the upper part of the roof being drier than the lower part of the roof? No, there really wasn't. So 397 was in our top position, 400 was in our bottom position, and you can see that we did not see sort of a drying trend from the top of the slope to the bottom of the slope. And so we concluded that because of these really uniform hydrologic properties on the roof, we didn't see any consistent patterns of spatial variation. All right, so this is there's Laura in the top left and me on the bottom, actually out in the field. Really hard picture to see, but this is an example of a vertical soil moisture array. So we took those 4 sensors from the green roof and we moved them to a bioretention cell on her field site. So you can see I got the job of jamming my hand down into the hole and then pushing the soil moisture sensors in horizontally in the hole. You can see where the water table is in the hole. You can't see very easily here, but this is the cable for those soil moisture sensors going down in. And so when the soil is saturated, soil moisture is just going to tell us the same thing. It's not going to tell us anything more than that, just that it's saturated. But of course, the water table position is going to move up and down in this bioretention cell over the course of the summer. So we tried to get some soil moisture sensors as deep as we could, even if we were below the water table at the time of installation. And this is what this data looks like. So this is from the summer of 2016. And so again, 397 is closest to the surface, followed by 398 and 399. And 400 is actually down in a gravel layer below the soils. So again, we see some responses to rainfall events. Of course, our biggest rainfall event happened when the weather station wasn't recording data, which was very frustrating. We see a really rapid response in the gravel layer with much smaller responses in the soil layers. And we see that after this big rainfall event, our deeper soils, or our sort of top and bottom soils wetted up and kind of stayed wet, whereas the middle part of the bioretention cell really didn't show too much response at all to those events. So we're still working on interpreting this data, but we're definitely seeing responsiveness in the soil moisture within these bioretention cells, which are designed to infiltrate water relatively quickly. And we definitely see water getting to the gravel layer very quickly. Okay, so using sort of a similar idea of actually measuring how quickly the subsurface or how well the subsurface conducts electricity or how resistive it is to electricity, there have been some cool applications that try to take a spatial approach to this. So using a geophysical technique called an electrical resistivity tomography survey, some researchers at Michigan State looked at the influence of vegetation on soil moisture patterns across a couple of different seasons, and they were contrasting a forested area and an open grass area. And so this is some data from the summer. And what they see here is the red colors are a low moisture content, and the green and blue colors are higher moisture content. So they see much lower water content in the soil below the forest than below the grasses. So why might that be? Well, it could be canopy interception, reducing the amount of rainfall that's even getting to the forest floor. And it could also be the greater water use associated with trees relative to grass. And so if you want to read the whole paper, you can read the whole paper that they published on this, and they show some differences between winter and summer. But one of the implications of this work is that when we think about either planting forests or converting forests to farmlands, we're going to change the whole water balance of the system because of these different soil moisture dynamics reflecting different amounts of plant water use. All right, so all of those things that I've just talked about in terms of measurement methods were based on electricity. Now for something completely different. So this is a neutron probe, so you can guess it's going to have something to do with radioactivity. And in fact, what you have is a radioactive source, some americium/beryllium there that is emitting fast neutrons into the soil. Those spark the release of neutrons from hydrogen, mostly from the soil water itself, to be emitted, and they come back and are detected here. And so what happens is that you place this neutron probe over a specially prepared, usually aluminum access tube, and then you lower it down to about 9 inches below the ground surface. The radioactivity is emitted. Of course, that's happening all the time. And then the detector counts for something like 16 seconds how much neutrons it's getting back from the slow neutrons, and that the higher the detection, the more the water content is. And then you lower the source tube down a little further, and you can repeat those measurements. So some of the advantages of these sort of techniques is that it measures a relatively large volume of the soil. So each measurement depth is going to measure the size of about a volleyball. And I didn't say this with the capacitance sensors that we use, but we're looking at sort of a 1 liter Coke bottle sort of size that we're measuring with those capacitance sensors. And some smaller capacitance sensors are measuring an even smaller volume of soil. So the neutron probe gives us a little bigger area to measure over. A disadvantage of this, as you might suspect, is that you now have a radioactive source material. So you have to be properly trained and licensed and maintain this and store this in correct ways. So these were very popular some time ago, like the 70s and 80s, but these days I see the TDR, the time domain reflectometry, and the capacitance sensors being far more widely used than neutron probes because they're cheaper and there are fewer risks associated with using them and fewer hassles. Another technique that we can use to measure water content is a tensiometer. So do you remember back when we were talking about plants and we talked about matrix potential, or the tension felt or suction felt in the plants? Well, we're going to talk about the same thing in terms of soils here in just a minute. And so the tensiometer is one of the few techniques we have for directly measuring the matrix potential or the suction in the soils. So the way this works is that you can see it's sort of got a vacuum gauge here and a long plastic tube filled with water, and then a porous tip down here. And so water is going to move out of the tube, or down the tube and out of the tip into the soil, because of course this is going to be inserted into a hole in the soil, snugly fit so that there isn't air around it. And so the rate at which water is sucked out of the tube by that matrix potential or suction in the soil is a function of how wet the soil is. So drier soils are going to suck harder than wetter soils. And no air can get into this tube. So the more water that's being sucked out, you're getting a vacuum at the top, and this vacuum gauge then can measure the tension. So you can put these tensiometers into particular locations at your field site and at different depths, and you can use them to measure the matrix potential and by proxy the water content of the soils. Again, you'd probably want to calibrate these tensiometers directly to volumetric water content if that's what you're interested in, but sometimes it's nice to get direct measurements of just that matrix potential itself. These typically would be installed at a single location for a field season. So if you want to measure matrix potential or water content at lots of locations, then you need lots of these. And in this example, you can see that it is manually read. So that's great if you just want data every few days or every time you're out in the field. A lot of times these are used for determining whether crops need to be irrigated or not. So you can see it's the Irrometer brand. But if you want data on, say, 5-minute timestamps, the tensiometer is going to be, in this configuration, is not going to work for you.